Hello everyone, I am Payal Priyadarshini, PGT Biotech from Kendriya Vidyalaya number 3, Delhi Cant. So today in the IIT PAL series of learning, we will learn about mutation, human genome project and DNA fingerprinting from the chapter Molecular Basis of Inheritance, class 12 textbook NCERT. So with the start of mutation, mutation is any type of change in DNA sequence or chromosome is called mutation. So mutation is if a DNA or a chromosome get changed by any way, that change will change the whole protein in many of the cases, and such type of changes in DNA sequences or chromosome is called mutation. Now here is a big question, what will be the effect of mutation? So as we know, in case of central dogma, which we learned in earlier session, central dogma is transfer of genetic information from DNA to RNA and from RNA to protein. So ultimately DNA is responsible to synthesize the protein, and that fragment of DNA which will code for the protein is called gene. So if anyhow there is a change in the gene or the fragments of DNA which will code for the messenger RNA or protein, will change the whole protein, and as we know, the most of the functions in our cells, in our body is performed by the protein. If the protein got changed, the function of protein will be altered and ultimately we will be having different types of disease. So mutation in functional part of DNA will lead to the different type of diseases. You might have learned about sickle cell anemia, which is caused by the mutation in hemoglobin gene. So there are so many examples which leads to the disease because of the mutation. Now types of mutation. Types of mutation on the basis of occurrence of the cell, there are two types. One is somatic mutation and another is germ line mutation. What do you know what is somatic cell? Yes, somatic cells are those cells which are responsible for the growth, repair and division of the cell in our body in the living system. So if any type of change in DNA or chromosome occurs in somatic cell, that type of mutation is called somatic mutation. And somatic mutation cannot be transferred from one generation to another. Second type of mutation on the basis of occurrence of cell is germ line mutation. Germ line cells are those cells which are responsible for gamete formation in the living system. So if there is change in germ line cells, it will lead to the change in the gametes, and accordingly it will be inherited to the another generation. So the generation to generation it will be passed out. Next type is point mutation. Point mutation means there is change in one or few base sequences of the DNA. As we know, DNA have three principal elements: sugar, nitrogenous base and sugar and phosphate. So all this sugar, phosphate and nitrogenous base combines together to form nucleotides, and nucleotides combines together to form DNA. So if any type of change occurs in one nucleotide or few nucleotides, it will lead to point mutation. There are two types of point mutation. One is base pair substitution and another is base pair insertion or deletion. Base pair substitution means there may be replacement of one nucleotide with the another nucleotide. If one base is being replaced by another base, that type of replacement is called base substitution mutation. And second type is base pair insertion or deletion. Base pair insertion or deletion means in the sequences of the DNA, any of the base pair will be inserted or it will be deleted from the DNA, and such type of mutation is called base pair insertion or deletion. So, here are some types of base pair substitution. One is transition mutation and another is transversion mutation. Transition mutation is when purine replaced by purine and pyrimidine replaced by pyrimidine. As we know, in DNA there are two types of nitrogenous bases, one is purine and another is pyrimidine. And in case of purine, in DNA there are two type of base, adenine and guanine. So, if in a particular DNA sequence adenine is replaced by guanine or guanine is replaced by adenine, means purine is replacing the another purine, or if we talk about pyrimidine, pyrimidine is again two type in case of DNA, thymine and cytosine. So, if thymine is being replaced by cytosine or cytosine replaced by thymine, pyrimidine replaced by another pyrimidine, such type of replacement is called transition mutation. That means purine replaced by purine and pyrimidine replaced by pyrimidine. Next type of base pair substitution mutation is transversion mutation, in which purine is replaced by pyrimidine and pyrimidine is replaced by purine. That means if a particular DNA sequence have either adenine or guanine, that will be replaced by either thymine or cytosine. That means purine will be replaced by pyrimidine and vice versa, pyrimidine will be replaced by purine, and such type of mutation is called transversion mutation. So, here are some examples of point mutation. The first one is missense mutation. Missense mutation is a gene mutation in which a base pair changes in a messenger RNA codon so that different amino acid is inserted into polypeptide chain. That means there will be a change in DNA and that will leads to the change in messenger RNA, and accordingly if there is change in messenger RNA, the amino acid coded by that messenger RNA will also get changed. So, such type of mutation in which a base sequence is changed and the change causes the change in the codon, and accordingly the amino acid coded by that codon will be changed. So, because of change in the amino acid, a different type of protein will be synthesized, and such type of mutation is called missense mutation. Here you can see in this particular DNA, here we have AAA and complementary TTT. Now, this TTT will form AAA messenger RNA which codes for lysine. If this AT base pair is being replaced by GC base pair, now this GAA complementary CTT, this CTT will form GAA in messenger RNA, and GAA will code for another amino acid, glutamine. So, the change in which a new amino acid is being coded by messenger RNA is called missense mutation. Can you identify which type of mutation is it? Transition or transversion? You can see adenine, which is a purine, and guanine, it is also a purine. It means purine is replaced by another purine, and this is transition mutation. Now, next example of point mutation is nonsense mutation. A nonsense mutation, it is a gene mutation in which a base pair change or alters an messenger RNA codon for an amino acid to a stop codon. That means, if there is a change in DNA sequence which will lead to the change in messenger RNA, and the messenger RNA will code for stop codon. As we know, we have 3 stop codons, UAG, UGA and UAA. So, here you can see in the slide, if in our original DNA we have AAA as triple codon, complementary is TTT, and the messenger RNA coded by this DNA will be having AAA, which will code for the lysine. If this AT base pair is replaced by TA base pair, here you can see now it become TAA, complementary ATT, and this ATT will form UAA codon in messenger RNA, and this UAA is one of the stop codon which will code for termination of translation. So, in case of nonsense mutation, the protein synthesis will be terminated there only. Next example of point mutation is neutral mutation. A gene mutation in which a base pair change a codon in the messenger RNA such that the resulting amino acid substitution produces no detectable change in the function of protein translated from that messenger RNA. That means, a change in DNA which will change the messenger RNA, and the messenger RNA will code for another amino acid, but the amino acid coded by that messenger RNA will be chemically equivalent to the original amino acid coded by the original messenger RNA. Here you can see in the slide, here we have again AAA, complementary TTT. This DNA will form AAA messenger RNA which code for lysine. This AT base pair is replaced by GC base pair. Now, here this TCT will form AGA messenger RNA, and here AGA will code for arginine amino acid. This lysine and arginine is chemically same, they are chemically equivalent. So, there will not be any change in the function or in the structure of the protein, and such type of mutation is called neutral mutation. Next type of point mutation is silent mutation. A gene mutation that changes a base pair, but the altered codon in the messenger RNA specifies the same amino acid in the protein. That means, if there is a change in DNA which will lead to the change in messenger RNA, but the changed messenger RNA will code for the same amino acid. As we know, one amino acid can be coded by more than one triplet codon. So, here you can see in the original DNA we have TTT in DNA. This TTT will form AAA sequences in the messenger RNA. Now, this AAA triplet codon will code for lysine. If this AT base pair is being replaced by GC base pair, here this TTC of DNA will form AAG messenger RNA triplet codon, and this AAG again code for the lysine. So, in silent mutation, there is change in the DNA sequences which will change the messenger RNA also, but the messenger RNA after change will code for the same amino acid, and this type of mutation is called silent mutation. Next example is Frameshift mutation, shifting of frame. That means if one or more base pairs are added or deleted from a protein coding gene, the reading frame of a messenger RNA can change downstream of the mutation. That means if there is insertional or deletion of any base pair from the original DNA, the whole triplet codon will be shifted. You can see in the picture, this is our original DNA. This strand of DNA will code for this messenger RNA, and this messenger RNA will code for this amino acid. Here in between this AA and TT base pair, there is insertion of GC base pair, and because of insertion of GC base pair, as we know, the codon are triplet codon. 3 base sequences of messenger RNA will code for one amino acid. So, starting from this UCU, you can see because of the insertion of GC, the whole messenger RNA codon is being shifted. Earlier, the original messenger RNA was coding for these amino acid, but because of the insertion of one base pair, the whole triplet codon got shifted, and because of the shifting in the sequences, there is change in the amino acid coded by the messenger RNA. So, the whole protein will get changed because of the insertion or deletion of any base pair in the original DNA. Now, another type of mutation on the basis of occurrence. There are 2 types of mutation on the basis of occurrence. One is a spontaneous mutation and another is induced mutation. A spontaneous mutation occurs naturally. Naturally means there is no any external factors which will induce the mutation in the DNA. It occurs naturally, and such type of mutation is called a spontaneous mutation. In case of induced mutation, it occurs when an organism is exposed to a physical or chemical agent called mutagen. So, a mutagen is a physical or any chemical agent which will cause the mutation, which will alter the sequences of the DNA, which will change the sequences of the DNA, is called mutagen. And if a mutagen induces the mutation in the DNA, that type of mutation is called induced mutation. So, a spontaneous mutation may be caused by DNA replication. You have already learned about this in DNA replication topic. During DNA replication, if anyhow DNA polymerase is added any wrong nucleotide in the newly synthesized DNA, at the same time the DNA polymerase will use its exonuclease activity. It will remove the wrong nucleotide and will add the correct one. So, at the same time that error, that damage, that mutation is being corrected. And second reason of a spontaneous mutation is chemical change. Chemical change may be depurination or deamination. Depurination means removal of purine nitrogenous base from the DNA, and deamination is removal of amine group from the base. So, such type of chemical change occurs naturally in the DNA, which will lead to the change in DNA, which will cause the spontaneous mutation. So, depurination is the loss of purine from the DNA when the bond hydrolyzes between base and the deoxyribose sugar, resulting in an apurinic site. That means, you know, in case of nucleotide, we have three principal chemical component. One is sugar, second one is nitrogenous base, and third one is phosphate. In case of DNA, we have deoxyribose sugar at the first number of carbon, here it is nitrogenous base. At the second, there is deficiency of one oxygen. At the third, here we have OH group, and at the fifth number of carbon, we have phosphate group. So, if this nitrogenous base is purine, purine means either adenine or guanine. So, if this purine is hydrolyzed, it will be removed from the sugar, and the remaining portion, that is phosphate and sugar, will considered as apurinic site. Apurinic site means there is no any purine at the first number of sugar in the DNA. You can see in this diagram, this is a guanine, nitrogenous base, sugar and phosphate. This guanine has been hydrolyzed with the water and get removed from the sugar. Now, this particular nitrogenous base has been removed, and this sequence is having only sugar and phosphate. Here at the first number of carbon, there is no any nitrogenous base, and this site is called apurinic site without any purine nitrogenous base. I hope depurination must be clear to you. Next, a spontaneous chemical change is deamination. Deamination means removal of amino acid from base or amine group from the base. You can see in case of cytosine, if there occurs a deamination, this cytosine will be converted into uracil, and this is the reason why DNA have thymine instead of uracil. You can see if there would not be thymine in DNA and uracil would be there, so it will be quite difficult to identify which one is the original uracil and which one has been converted from cytosine. So, DNA is having thymine instead of uracil because of this particular reason. In the deamination of cytosine, it will converted into uracil, and if 5-methyl cytosine will undergo deamination, it will convert into thymine. So, such type of deamination occurs in nitrogenous sequences spontaneously, naturally, which will lead to the spontaneous mutation. Now, induced mutation. As we learned earlier, induced mutation is caused by certain external factors, and that factors are called mutagen. Mutagen may be physical like radiation, UV rays, X-rays are common example of radiation which causes the mutation, and mutagen may be chemical. In case of chemical, there are so many examples. One is base analog, base modifying agent and intercalating agent. base analogues are chemically and structurally similar nitrogenous bases. Base modifying agents are those chemicals which will modify the nitrogenous bases, and intercalating agents are those chemicals which will insert itself inside the base sequences of the DNA. Base analogues are those bases that are similar to those normally found in DNA. So, in cell there are certain chemical compound which are structurally similar to the normal nitrogenous bases which is found in the DNA. One of the example of base analog is 5-bromouracil, which is a base analog of thymine. If we can see in the slide, the thymine and 5-bromouracil, there is a single change. Here we have CH3 methyl group in thymine, and in case of 5-bromouracil, instead of methyl group it is having bromine. So, structurally both these chemical compounds are same, and if anyhow this 5-bromouracil is being inserted inside the DNA instead of thymine, it will lead to the change, it will lead to the mutation in DNA. So, base analog are those chemicals which are structurally similar with the normal bases. Next is base modifying agent. You can see the name base modifying agent. The agent, the chemical which can modify the structure of bases. So, base modifying agents are the chemicals that act as mutagen by modifying the chemical structure and property of base. So, base modifying agents, it is one of the chemical mutagen which can change the structure of normal bases, and accordingly because of the change in the structure, the property of base can also get changed. Here are some examples of base modifying agent. One is nitrous acid, which will lead to deamination. Deamination means removal of amine group from the nitrogenous base, which we learned earlier in this session. Hydroxylamine, this hydroxylamine can add the hydroxyl group in the nitrogenous bases. Methylmethane sulphonate, it is one of the alkylating agent. Alkylating agent means which adds the alkyl group in the nitrogenous bases. So, these nitrogenous bases can be modified by certain base modifying agent, and these base modifying agents are nitrous acid, hydroxylamine and methylmethane sulphonate. There are so many another examples also of base modifying agent which can change the structure of the normal bases. Another chemical mutagen which induce the mutation in the DNA is intercalating agent. These intercalating agent insert themselves between adjacent bases in one or more strands of the DNA duplex. So, intercalating agents are those chemicals which can insert themselves in between the basis sequences of the DNA, and because of the insertion the whole DNA sequences will get changed. Here are some examples of intercalating agent: proflavin, acridin, ethidium bromide. So, these chemical compounds can naturally get inserted inside the DNA sequences and can change the whole DNA sequences, and accordingly the messenger RNA will get changed, and if messenger RNA will get changed, the protein will get also changed, which can lead to the different type of diseases. So, these are certain examples of chemical mutagen. Now, chromosomal mutation. Chromosomal mutation may be change in the chromosome structure, number or set. That means, if there is change in the number of chromosome or there is change in the structure of chromosome or there is change in the set of the chromosome, all these type of changes in the chromosome will lead to the chromosomal mutation. Now, here are some examples, some of the types of change in chromosomal structure. One is deletion, second one is insertion, third one is duplication, fourth one is inversion and fifth one is translocation. These are some of the types of chromosomal mutation which leads to the change in chromosome structure. Deletion means if certain fragments of chromosome get deleted from the chromosome, it will lead to the change in the chromosome structure. Insertion means certain fragments will get inserted inside the chromosome, which will change the structure of the chromosome. Duplication means there is formation of duplicate fragments of the chromosome which will lead to the change in the structure. Inversion means certain fragments of DNA in the chromosome will get rotated by 180 degrees Celsius, and because of that the whole sequences will be inverted, and this inversion will lead to the change in chromosome structure. Translocation means certain fragment of DNA will change its position and will transfer from one place to another. It can also lead to the change in structure of chromosome and will cause the chromosomal mutation. Now, variation in chromosomal number. Chromosomal number can be increased or decreased depending on the condition or situation. When an organism or a cell has one complete set of chromosome or an exact multiple of complete sets, that organism or cell is said to be euploid. So, euploid means when a cell or an organism have complete set of chromosome. As we know, the human beings are diploid in nature. So, we have 2n set of chromosome, and this complete set of chromosome for any organism is called euploid. Chromosome mutation that result in variation in the number of chromosome set occurs in nature, and the resulting organism or cell are also euploid. Chromosome mutation resulting in variation in the number of individual chromosomes are examples of aneuploid. So, if there is change in the number of chromosome or set of chromosome, it will lead to the aneuploidy of the chromosome. So, here are some type of aneuploidy in which certain numbers of chromosome will get changed. The one is nullisomy. Nullisomy means a loss of one homologous chromosome pair. That means we know in case of human being, a single cell have 46 chromosome, and this 46 chromosome contains two set of chromosome which is denoted as 2n, and this complete set of chromosome is called euploid. So, in case of human being, 23 plus 23 23 chromosome from the mother, 23 chromosome from the father, will give you 46 chromosome. And if this 46 chromosome get increased by number or decreased by number, it can lead to the change in chromosome number. And if 46 chromosome reduced to 2 and only 44 chromosomes will remain in the cell because of deletion of, because of removal of one complete pair of the chromosome, this is called as nullisomy. So, nullisomy is the loss of one complete chromosomal pair, that is 2 chromosome. So, out of 46, when 2 chromosome will get removed or lost, it will lead to the aneuploidy, that is called nullisomy. So, in case of nullisomic cell or organism, there will be 44 chromosomes in a cell. Second type of aneuploidy is monosomy. In case of monosomy, it involves a loss of single chromosome. So, as we know, the cell is having 46 chromosome in case of human being, and if there is loss of just one chromosome, it will lead to the 45 chromosome, and this type of mutation in chromosomal number is called monosomy. So, in monosomic cell or organism, there is just 45 chromosome in their cell. There is loss of one chromosome from the set. Next example of aneuploidy is trisomy. In case of trisomy, the 46 chromosome will increase in one chromosome number, which will lead to 47 chromosome, and a cell, an organism, if they have 46 chromosome in their cell, that will call as trisomy. So, trisomy are those organisms or those cells in which there is 47 chromosome in a cell. That means there is increase in number of one chromosome. So, 46 plus 1 will become 47. Next example of aneuploidy is tetrasomy. Tetrasomy, in case of tetrasomy, this 46 chromosome will increase by number of 48. That means there is increase in 2 chromosome in the complete set of chromosome, and there will be 48 chromosome, which will lead to tetrasomy. So, a tetrasomic cell or organism have 48 chromosome instead of 46 because of increase in one pair of chromosome or 2 number of chromosome. So, 46 plus 2, it will become 48, and this 48 chromosome is there in tetrasomic organism or cell. Changes in complete set of chromosome. Till now we learned about changes in the structure and changes in the number of chromosome, but there will be a change in the complete set of chromosome. There are two type of changes in set of chromosome. One is monoploidy and second is polyploidy. In case of monoploidy, a monoploid individual has only one set of chromosome instead of the usual 2 sets. So, normally all the living organisms have 2 set of chromosome, which is denoted as 2n, and if there is loss of one complete set from there, it will lead to the only one set of chromosome, and such type of organism or cell is called monoploidy. So, monoploidy organism or cell in which only one set of chromosome is there because of loss of one set of chromosome from the complete set of chromosome. Next is polyploidy. Polyploidy is the chromosomal constitution of a cell or an organism that has more than the normal two sets of homologous chromosome. That means, as we know, normally there is 2n set of chromosome. If this 2 set is increased by 1 set or 2 set or accordingly 3 set, 4 set like that. So, it can lead to 3n cell, 4n cell, 5n cell, 6n cell. So, such type of changes in set of chromosome is called polyploidy. Such type of chromosomal mutation found mainly in the plants. So, monoploidy is the decrease in the one complete set of the cell and polyploidy is the increase in complete set of chromosome in the cell or organism. So, I hope the mutation would be clear to you. Now, you must be knowing that all the time there occurs a mutation in the DNA. Whenever the replication is there, whenever we are exposed to certain chemicals compound, but there are certain enzymes which involve in the repairing of the DNA damage. That means mutation would be there, but that mutation will be corrected, repaired by the enzymes which is found, which is present in our cells. And there are two type of repair mechanism of mutation. One is direct repair system and another is excision repair system. So, in our cells, in our body, the mutation is corrected, the mutation is repaired naturally by the enzyme synthesized by our genes. But you should know that only less than 2% of our DNA is functional. So, most of the time the mutation occurs in non-functional DNA sequences which cannot lead to any change in the protein. But if anyhow functional DNA is being changed or mutated, it can lead to the change in the protein and ultimately we can have different type of disease. But it does not matter if mutation occurs, that will be corrected by certain enzymes. In case of direct repair system, the mutation, the damage is reversed at the same time, only the damaged portion is corrected, and in case of excision repair system, along with the damaged portion, certain normal fragments of DNA get also removed from the DNA and then the gap of the DNA will be covered up or replicated by DNA polymerases. So, direct repair system having different type of mechanism or different type of enzymes. The first is mismatch repair. In case of mismatch repair, DNA polymerase proofreading is there, which we already learned in DNA replication chapter. If there is incorrect nitrogenous bases is being added up by the DNA polymerase, at the same time DNA polymerase will remove the wrong one and will add the correct one. So, there is correction of only wrong nucleotide or damaged nucleotide, that is why it is considered as direct repair system. Another example is repair of ultraviolet induced pyrimidine dimers, especially thymine dimers, by photolyase enzyme which is encoded by PHR gene. So, if we are exposed to ultraviolet rays, it will lead to the thymine dimer formation, and that type of thymine dimer formation can be corrected by an enzyme called photolyase, and this photolyase enzyme is encoded by a gene which is called PHR gene. This repair system is also called as light repair system because this photolysis enzyme is coded in the presence of light. Another example of direct repair system is repair of alkylation damage by O6 methylguanine methyltransferase enzyme, which is encoded by ADA gene. So, if there is certain methyl group found in nitrogenous base which were added by some base modifying agents, alkylating agents, such type of mutation is being corrected by O6 methylguanine methyltransferase enzyme, which is encoded by ADA gene. So, these are some of the examples of direct repair system in which only the damaged portion is being corrected. Excision repair. Excision repair means the fragments of DNA which have the damage is being removed along with the normal fragments, and then the gap is filled up by DNA polymerase, and finally the nick is being sealed up by the DNA ligase. So, here we have certain methods, certain different mode of repair system in case of excision repair. The one is base excision repair. Base excision repair is mainly for depurination and deamination mutation, and such type of mutation is repaired by glycosylase enzyme. This glycosylase enzyme will remove the damaged portion along with the normal one, and then that gap is filled up. Next example of excision repair system is nucleotide excision repair. This nucleotide excision repair is corrected by enzymes UVRA, UVRB, UVRC and UVRD, which are coded by UVRA, UVRB, UVRC and UVRD genes respectively. So, these enzymes involved in nucleotide excision repair. Another example, another method of excision repair is methyl directed mismatch repair, which is encoded by MutL, MutS and MutH enzymes, and these enzymes are synthesized by MutL, MutS and MutH genes respectively. So, all these enzymes, all these genes involved in excision repair system. In case of excision repair system, you must understand certain step which is common in all. The first step is identification of damaged portion. Second step is removal of damaged DNA along with correct sequences. Third step is filling of gap by DNA polymerase, and fourth step is sealing of nick by DNA ligase enzyme. So, these are four common steps for excision repair, whether it is base excision repair or nucleotide excision repair or methyl directed mismatch repair. In all these excision repair system, these are the common steps for excision repair. That means, if suppose here we have a DNA and this particular fragment is damaged, this is the damaged portion of DNA. This is our DNA. So, first of all, the enzymes will identify this damaged DNA and will add it over there. After the identification, these damaged portion will be removed along with the correct one. Now this gap will be filled up by DNA polymerase. And now this nick will be sealed up by DNA ligase, and now the whole DNA will be corrected. So, you can see these are the some common steps for excision repair system. First, the damage will be identified by the enzymes. Now, along with that damage, correct sequences are also get removed up. The removal of that damaged portion along with the correct one will give you the gap in the DNA. These gaps are filled up by the DNA polymerase. After the filling of gap, final phosphodiester bond is not being created, it will create a nick. This is called nick. Now this nick is being sealed up by DNA ligase. So that is how the damage is being corrected by excision repair system. Now let us answer this question, what is mutation? Yes, your answer is right. Mutation is any type of change in DNA or chromosome. What will be the effect of mutation on living system? If mutation is there in the functional part of DNA, it will lead to the change in the protein, and because of change in the protein, it will lead to the different type of diseases. But if mutation is there in the non-functional DNA, it will never be passed to the messenger RNA because it is a non-functional DNA and there would not be any change in the protein. So, if mutation is there in the non-functional DNA, there would not be possibility of getting any disease. What is trisomy? Trisomy is a increase in one number of chromosome in a cell or in any living organism. If a particular cell or any organism having 46 number of chromosome, and if one more chromosome is being added up, the complete number will be 47, and this will lead to the trisomy. Define mutagen. Mutagen is an external factor which leads to the induced mutation in the DNA. The mutagen may be physical like radiation, UV rays and X-rays, and the mutagen may be chemical. The chemicals like base analogues which are structurally similar to the normal bases, base modifying agent which can change the structure of the bases, and intercalating agents which insert itself inside the normal sequences of the DNA. So, mutagens are the factors which cause the mutation in the DNA. Do you think mutation can be repaired? Yes, mutation is repaired by many of the enzymes synthesized in our cells. The repair may be direct or it may be excision. In case of direct repair, only the mutation is being corrected, and in case of excision, the mutated region along with the correct sequences are being removed up, and the gap is being filled up by DNA polymerase and the nick is being sealed up by the DNA ligase. So, mutation can be corrected, can be repaired by our enzymes synthesized by our cells. So, I hope this session on the topic mutation must be clear to you. In the next session, we will continue for another topic, Human Genome Project and DNA fingerprinting. Till then, thank you. Have a nice day.